Today we have discussed about the fundamental unit of life. Our earth is inhabited by millions of different kind of living organisms. These living organisms are bacteria, fungi, plants and animals. Although the great diversity of organisms look very different forms of other, but all these organisms exhibit a unique similarity in their organization. Can you guess the structure which is common to all these organisms? Yes, it is cell. The bodies of all living organisms are made up of microscopic unit called cells. Each of us has about 50 million cells, an enormous number which is difficult to image. Thus, cell is the basic structural and functional unit of living organism. Cell biology is a study of cell in all aspects of structure and functions. What is cell? All organisms are composed of cells. Some are composed of a single cell and are called unicellular organism, while other like as composed of many cells are called as multicellular organism. Unicellular organisms are capable of independent existing and performing the essential function of life. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is the fundamental structure and functional unit of all living organisms. First cell discovered by Robert Hooke in Cork. Discovery of the cell. In Robert Hooke, 1665, an English scientist saw cells for the first time in a thin slice of cork with its microscope. He observed and described the cells as honeycomb-like structure. He named the box-like compartments as cellulae or cell. The term cells from Latin cella means little room or hollow shapes. In 1674, Anton van Leeuwenhoek, a Dutch scientist, studied living cells for the first time with the help of an improved microscope, which he himself had made, though he was the first one to observe cells, but he did not use the term cell. In 1838, two biologists, J.M. Sliden and T. Swan, proposed the cell theory. According to them, the cell is the structural and functional unit of living beings. In 1855, Rudolf Virchow, a German biologist or pathologist, proposed that all the cells arise from the pre-existing cells. He studied his Latin as omes, cellulae, cellula. Cell theory therefore states that all living organisms are composed of one or more cells. The cells is the basic unit of life. Cell developed from pre-existing cells. The cell theory, Slade and Swan studied a different type of animal cells and reported that cells had their outer layer, which is today known as the plasma membrane. He also concluded, based on his study on the plant tissues, in the presence of cell wall is the unique characters of the plant cell. Swan proposed the hypothesis that the body of animals and plants are composed of cells and product of cells. Slide and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Slide and Swan gave the cell theory a final shape. Cell theory was understood today as All living organisms are composed of cells and product of cells. All the cells arise from pre-existing cells. An overview of cells. The onion cell, which is a typical plant cell, has a distinct cell wall and its outer boundary, and just within it is the cell membrane. Cells that have a membrane-bounded nuclei are called eukaryotic, whereas cells that lack a membrane-bounded nucleus are prokaryotes. In both the prokaryote and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of cell. The cytoplasm is the main area of cellular activity in both plants and animal cells. Various chemical reactions occur in it to keep the cell in living state. Besides the nucleus, the eukaryotic cells have other membrane-bounded distinct structure called organelles like endoplasmic reticulum, the Golgi apparatus, lysosome, mitochondria, microbodies. The prokaryotic cells lack such membrane-bounded organelles. Ribosomes are non-membrane bound organelles found in all cells. Both eukaryotic cells as well as prokaryotic cells within the cell ribosome are found not only in the cytoplasm but also within the two organelles chloroplast and mitochondria and on rough endoplasmic reticulum. Animal cells contain the another non-membrane bound organelles called centriole with help in cell divisions. Next, cell shape, size and numbers. Look at the structure of onion peel cell. Do all cells look alike in shape and size? Yes, all cells are some shape and almost of small size. The onion peel cells containing for nucleus and dense cytoplasms. Let us have look like at cells from the different part of the plant body. Now are all cells are the same? No, they are not. Palisade leaf cells containing for chloroplast, large vacuole and nucleus, and stomata containing for two guard cells and subsidiary cells. The xylem elements also difference. Now, if we look like at the cells from different part of human body, they too do not look alike in shape and size. As you can see the cell is different in shape and size. The human sperm cells, head, tail and in between body system also is there. The epithelial cells and cartilage cells, smooth muscles fibers, liver cells, fat cell and stained muscles fibers, the nerve cells. Plant cells are different from animal cells. Plant cells in a root are different to those in the stem or in the leaf. Animal cells, including cells in our body, are all sort of different shape and size. It means cell vary in number, shape, size in different organism and also in different part of same organism. In fact, the shape and size of the cells are related to function they perform. For example, in plant cell, xylem and phloem are elongated in shape and helps in conduction of waters and food. Similarly, in animals, nerve cells are long and branched that helps in conduction of information from one part of body to another part. Cell size. The size of the cell range between the broad limits. Some cells are extremely small and can be seen only when they are magnified and it visible only with a microscopic sense. They are only a few
micrometers in diameters. Cells are limited in size by the ratio between their outer surface area and their volumes. Small cells have more surface area for their volume of cytoplasm than large cells. As cell grow the size, grow the amount of surface area become the tool small to allow material to enter and leaves the cell quickly enough. The cell size is also limited by the amount of cytoplasmic activities that the cell nucleus cannot control. Mycoplasma, the smallest cell, only 0.3 micron in length, that is called as a PPLO. Pleuropneumonia-like organism is an example of mycoplasma having the size about 0.1 micron. Bacteria 3 to 5 micron. The largest isolated single cell egg of an ostrich. Human red blood cells 7.0 micron in diameters. Nerve cells is the longest cell. Followed by the cell shape. The shape of the cell of the cells is more variable in the size. The shape of the cells may be spherical, oval, elliptical, and spindle, polygonal, or flat plate-like. The shape of the cells is determined by the specific function of the cell. Some of the cells, like amoeba and white blood cells, can change their shape, while some of the plant cell and animal cells have almost fixed in shape. Among unicellular organisms, for example in amoeba, the shape of the cell is irregular, while among the multicellular organisms, like in plant, the xylem and phloem elements are elongated tubular structure so as to help it conduction of waters and minerals. The dividing meristematic cells are isodiametric in shape. Similarly, in animal cells, the nerve cells are elongated thread-like which enable an efficient communication system. The muscle cells are elongated and cortical that helps in movements of bone, while the red blood cells are biconcave shape. This kind of cells or shape of RBC helps the RBC easily move through capillaries and thus permit gaseous exchanging. Some example of animal cells: the nerve cells are neuron, and muscle cells, epithelial cell, red blood cells, and white blood cells. Some example of plant cell: epidermal cells, the barrel-shaped cells; xylem cells, the tube-like cells; phloem cells, sieve tube cells; and photosynthetic cells, that's called as a mesophyll cells of leaf. Cell volume. The volume of cell is consisting of particular cell type. It is independent of the size of the organism. For example, kidney or pancreatic cells are the amount of same size as in dog or cat. The difference in total mass of the organism or organ dependent on the number, not on the volume of the cell. Cell number. The number of cells varies in all living organisms based on the number of cells. The organism are divided into two types: unicellular organism, single-celled organism. The organism made up of single cells are called unicellular organism. Amoeba, paramecium, bacteria, etc. are example of unicellular organism. The single cell have the ability to perform all the life process like digestion, respiration, excretion, growth, and reproduction. In these cells, there is no division of labor as the single cells have to perform all the activities. See the bacteria structure. The flagella also is there, and pilus also is there, and nuclear materials also presented. The multicellular organism The organism made up of number of cells are called multicellular organism. Fungi, plants and animals are example of multicellular organism. All the cells of multicellular organism have a similar basic structure and similar base life activities. These cells group together to produce different tissues. Tissues from the organ, and the organs gives rise to organ system that perform specialized function. For example, an elephant, a tree. The increasing of order of complexity in the multicellular organism is cell, tissue, organ, organ system and organism. Just like that, a single cell is producing multiple tissues and organ, so just like that is a stomach, the organ system, that's called as a digestive system, the organism, that's called as a young baby. The difference between the unicellular organism and multicellular organism. The unicellular organism, the organism made up of a single cell are called unicellular organism. The multicellular organism, the organism made up of large number of cells are called multicellular organism. The unicellular organism, there is no division of labor. A single cell perform all the activities of organism. The multicellular organism, there is division of labor. Cells are specialized to perform different function. Unicellular organism, the lifespan of the organism is short. The multicellular organism, the lifespan of multicellular organism is long. Unicellular organism example, amoeba, paramecium, bacteria. Multicellular organism example, fungi, plants and animals. Structural organization of a cell. A cell is the basis unit of life. It performs all the basic life function like intake of food, excretion, metabolism, respiration, growth and reproduction. However, there is no mixing up of different function. In fact, there is a division of labor. For this, a cell usually possesses a number of components called cell organelles. Each cell organelles performs specific functions. Hence, a cell may be defined as a unit of protoplasm bounded by a cell membrane or plasma membrane. Cell membrane or plasma membrane or biomembrane. It is the outermost, extremely delicate, elastic membranes covering of each cell. It is separated the cytoplasm of the cell from its environment. Plasma membrane is a living, selectively permeable membrane. It allows some selected materials to move in and out of the cell and prevent the entry and exit of the other substances. The plasma membrane is made up of a bilayer of lipid and proteins. Small carbohydrates are attached at place to outer surface of lipid and proteins. See the plasma membrane. Phospholipid molecules and protein phosphate head, the membrane, the lipid tails also is there, the in between pore also is occur. Several models have been proposed regarding the arrangements of proteins and lipids. The fluid mosaic model proposed by Zinger and Nicholson is widely accepted. According to this model, the cell membrane is composed of lipid bilayer of phospholipids molecule into which variety of globular protein are embedded. Each phospholipid molecule has two ended, an outer head hydrophilic and in the inner tail hydrophobic. Protein molecules are arranged into two ways. Peripheral protein or extinct proteins are presented on the outer and inner surface of lipid bilayer, while integral protein or instinct proteins are embedded inside the lipid bilayer partially or wholly. Right. The fluid mosaic model. See, just like this, a plasma membrane, peripheral proteins also is there, the carbohydrate also is there, and glycoprotein also is there. The integral proteins and cholesterols also mixing there, phosphoglyceride molecules, and present for the cytoplasms. Function of the cell membrane. It provides a definite shape of the cells. It functions as mechanical barriers between the external and the internal environment of the cell. It regulates the movements of molecule in and out of the cells. The flexibility of the membrane helps the cell to engulf food and other substances from its external environment by endocytosis. For example, amoeba obtain food by endocytosis. Endocytosis in the ingestions of material by the cells through plasma membranes. Plasma membrane or cell membrane. The membrane acts as a physical barrier between cells and its surrounding environment. There are four types of membrane. Impermeable membrane that does not allow the substance to pass through. It is known as the impermeable. Permeable, the membrane that allow all kind of material to pass through. It is known as the permeable membrane. Semi-permeable, the membrane that is permeable to solvent but prevent the passage of solute, is known as a semi-permeable membrane. Selective permeable membrane, membrane that allow the pass of solvent and some selected solute, is known as a selective permeable membrane. Plasma membrane is a selective permeable membrane. Transport across the membrane. Passive transport. The transport of substance across the plasma membrane without expenditure of energy is known as passive transport. It is known of two types, diffusion and osmosis. Diffusion, the process of movement of substance from the region of higher water concentration to the region of lower concentration so as to spread the substance uniformly in the given space, is known as the diffusion. This process does not require energy. For example, transport of carbon dioxide and oxygen across the membrane. The gas like carbon dioxide and oxygen move across the membrane through diffusion. This process of respiration produce carbon dioxide inside the cell. As the concentration of CO2 increases inside the cell as compared to the outside, CO2 diffuse out of the cell into external medium. Similarly, the concentration of oxygen is always higher in the external medium as compared to the cell. Therefore, oxygen diffuses from outside to the inside of the cells. See the picture. The higher concentration move to the low concentrations. Osmosis. It is defined as the diffusion of water from region of higher concentration to region of low concentration across the semi-permeable membrane. This kind of movement is also concentration gradient. There is no expenditure of energy. For example, movement of water across the selective permeable membranes. For example, low concentration sugar solution and high sugar concentration solution. Just see the pictures. The transport of the selective permeable membrane. The difference between the diffusion and osmosis. Diffusion, it is the movement of substances from the region of higher concentration to the lower concentration. Osmosis, it is the movement of water from the region of higher concentration to its lower concentration through semi-permeable membrane. Diffusion, it takes place in the three medium, solid, liquid and gases. Osmosis, it takes place only in the liquid medium. Diffusion, it does not involve the semi-permeable membrane. Osmosis, it requires semi-permeable membrane. Diffusion, it is not much influenced by the presence of other substances. In osmosis, it is influenced by the concentration and type of solute particles. Type of osmosis. Endosmosis. Endosmosis is the osmotic entry of water into the cell. Exosmosis. Exosmosis is the osmotic withdrawal of the water from the cell. There are three types of osmotic solution which can cause osmosis across a semi-permeable membrane. Hypotonic solution. Hypo means less than or lower. Hypotonic solution which has lower osmotic concentration than the cell. See the picture, hypertonic solution and hypotonic solution. Low solute is concentration is go to outside, high solute concentration go to inside. Hypertonic solution. Hyper means more or higher. Hypertonic is a solute that has higher water concentration than the cell. Isotonic solution. Iso means same. It is the solution that has same osmotic concentration and inside the cells. Active transport. Active transport is the process of transport of molecule across the plasma membrane against the concentration gradients. The process required the use of energy. For active transport, the cell membrane passes ATP mediated carrier proteins. Glucose, amino acids and ions pass through the plasma membrane by active transport. Cell wall. Cell wall was discovered by Robert Hooke. Outermost but dead boundary of the plant cell is cell wall. Bacteria including plants based they have the cell wall. Cell wall is a rigid non-living covering present outside of the plasma membrane in plant cell. Animal cell lack of cell wall. Plant cell is made up of cellulose which is permeable to water, solute and gases. In fungi, the cell wall is made up of chain, a cementing layer called as middle lamella is present between the walls of two adjacent cells. Middle lamella is formed of pectin, calcium and magnesium pectate. See the picture. The middle lamella, primary cell wall and plasma membrane. The pectin and cross-linked glycan, cellulose microfibrils. Difference between the cell wall and cell membrane. Cell wall, it is found only in plant cells. Cell membrane, it is found in both plant and animal cells. Cell wall, it is the outermost layer of the plant cell. Cell membrane, it is lie on the outside of the animal cell and inner of the cell wall in plant cell. Cell wall, it is a thick and rigid. Cell membrane, it is thin and flexible. Cell wall, it is permeable. Cell membrane, it is selective permeable. Cell wall, it is formed cellulose and pectin. Cell membrane, it is formed of lipids and proteins. Cell wall, it provide protection and strength of the cells. Cell membrane, it acts as a mechanical barrier and regulate the movements of molecule in and out of the cells. Function of the cell wall. It gives a different shape of the cells. It provides mechanical strength of the plants. It protects the cell against the mechanical injury and pathogens. It helps in transport of various substances across it. Cell wall help the plant cell to withstand a lot of variations in the surrounding environments. Cell wall prevent the bursting of cell on endosmosis as it is quick, thick and rigid. The cell membrane, middle lamella is consist of calcium and magnesium. Amount of calcium is more. Fruits become soft and juicy due to the dissolve of middle lamella. Cellulose is a main constituent of cell wall, but addition of cellulose, hemicellulose, cutin, pectin, lignin, suberin all presented in cell wall. Cell wall worked as a frame of protective layer of cells. Cellulose is a microfibril and microfibril arranged in the layer to form of the skeleton of cell wall. In between this layer, other substances like pectin, hemicellulose may be presented. These form matrix of cell wall. Network of the cellulose fiber from the skeleton of the cell wall. 35 to 100 cellulose chain is 1 micelle, 20 micelle is equal to 1 microfibril, 250 microfibril is equal to 1 microfibril in cell wall. Middle lamella is a cement material between the two adjacent cells in multicellular plants or outermost layer of the cell wall. A difference between the primary cell wall and secondary cell wall. Primary cell wall, cellulose microfibril are arranged in the dispersed manners. Secondary cell wall, microfibril are parallel to long axis of cells. The primary wall, hemicellulose more than 50%, secondary cell wall, hemicellulose less than 25%. Primary wall, the primary wall has a lipid 5 to 10% and protein 5%. Protein, lipids, either absence or little amounts. Primary wall formed by interceptions. Secondary wall by both methods. The primary wall is universal layer, absence in meristem cells in the secondary wall. Nucleus. First of all, Leeuwenhoek observed nucleus in RBC of fish. Detailed study in arched root cells and named by Robert Brown in 1831. Credit of discovery goes to Robert Brown. Nucleus is a double membrane bounded design protoplasmic body which control all cellular metabolism and enclose genetic information of cell. Nucleus is considered as controller or director of cell. Importance of nucleus is control of heredity, growth and metabolism was experimentally proved by Hammerling. If the nucleus of a cell is experimentally removed, the unicellular organism will die after some time. Thus, the nucleus is very important and largest components of cell. Strasberg stated that nucleus arises from the division of pre-existing nucleus only. The study of nucleus is known as karyogamy. Generally, eukaryotic cells containing at least one nucleus, but nucleus is absent in mature phloem sieve tube elements and mature RBCs of mammals. Dikaryotic and multikaryotic cells are also known as multinucleated cells may be following types. Phenotypic cells. This type of cells are formed by the free nuclear divisions. Example, fungi, endospore, rhizopus and Boucheria of algae. Syncytium. Syncytium is formed by the fusion of cells. Example, epidermis of nematodes, striped mussels. Structure of nucleus. Nuclear membrane or nuclear envelope or karyotheca. Nucleoplasm or karyoplasm or karyolymph. Chromatin net. Nucleus or little nucleus or ribosome factory. Nuclear envelope. It is a double membrane structure that separated the nucleus from the cytoplasm. It is mainly made up of protein and lipids. The membrane is perforated by several nuclear pores which allow exchanging of material between the nucleus and cytoplasm. On the basis of presence or absence of nuclear membrane, organisms are divided into two types. One is a prokaryote. Pro means primitive, karyotic nucleus. Organism whose cells do not possess a well-formed nucleus is known as the prokaryotes. Example, bacteria and cyanobacteria. Prokaryotics also lack a membrane-bounded cell organelles. The cell organelles like mitochondria, Golgi body, endoplasmic reticulum are lacking in prokaryotes. Eukaryotes. Eu means true, karyotes mean nucleus. Organism whose cell possesses a nuclear membrane is known as eukaryotes. Cell organelles like mitochondria, Golgi body, endoplasmic reticulum are presented in eukaryotes. Example, plants and animal cells. A difference between prokaryotes and eukaryotes. Prokaryotes, an organized nucleus is absent. Hereditary materials lie free in cytoplasm and is known as nucleoid. A nuclear membrane is absent. In eukaryote, a well-organized nucleus is presented. It contains hereditary materials covered by a nuclear membrane. In prokaryotes, a single chromosome is presented and is represented by a single circular DNA only. That is called as a plasmid. In eukaryotes, number of rod-like chromosomes with two or several DNA molecules are presented. In prokaryote, membrane-bounded cell organelles are absent. In eukaryote, membrane-bounded cell organelles are presented. In prokaryotes, no compartments are found in prokaryotes. Distinct compartments in the cells, that is cytoplasm and nucleus, is present in eukaryotes. Example of prokaryotes, bacteria and cyanobacteria. Example include plants and animals, fungi in eukaryotes. See the picture. The bacteria containing of nucleoids, the outermost covering layer called as a capsule. The flagellum also is there. Cell wall also presented, and cell membrane, the ribosome. 70S ribosome are presented in the prokaryotes. In eukaryote, the nucleus is presented. The mitochondria also presented. Endoplasmic reticulum also presented. The 80S ribosome also presented. Nucleoplasm, otherwise called as a nuclear sac. It is colorless cytoplasm within the nuclear membrane in which chromatin and nucleoid are suspended. Nucleolus, it is dense, spherical, a granular structure found inside the nucleus. Nucleolus is not bounded by a membrane. It is a membraneless. It is rich in protein and RNA. Basically, nucleoid is a site of ribosome formations. Ribosome is torn, help in the protein synthesis in the cytoplasm. Chromatin network. Chromatin is a network of the fine thread-like coiled filaments uniformly distributed in the nucleoplasms. It is usually made up of DNA and protein. During the cell division, chromatin became a high, condensed, thick and rod-like structure known as chromosome. The chromosome containing genes which are composed of DNA. A gene is a functional unit of chromosome. These are arranged in a single linear order along the chromosome. One gene is responsible for the single characters, or a single characteristics may be transmitted by a set of genes. Genes are responsible for sorting and transmitting hereditary characters from one generation to another generation. See the structure of nucleus. The nuclear pores is there and nuclear envelope is there. Nucleolus also presented. The chromatin network also presented. Chromatin net, term given by Fleming. It is a intracellular long thread-like fine fiber which embedded nucleoplasm. Chromatin net is mainly formed a DNA and histone protein complex. Chromatin fibers containing genetic information and contents to form constant number of chromosomes during the cell division. Chemically, chromatin consists of DNA 31%, RNA 2 to 5%, histoprotein 36%, and non-histone 28%. 20 to 30% part of histone is made up of arginine and lysine amino acid. On the basis of relative amount of arginine and lysine, there are 5 types of histone proteins: H2A, H2B, H3, H4 and H1. Amino acid, lysine rich type of histone H. Amino acid, slightly lysine rich type of histone H2A, H2B. Amino acid, arginine rich type of histone H3 and H4. Chromatin net has 2 types of chromatins by Emil Heitz. Euchromatins. This is lightly staining and diffused a part of chromatin which is transcriptionally or genetically more active. Generally, euchromatins lie a central part of nucleus. Heterochromatin, this is a dark stained, thick and condensed part of chromatin. This part have more histone and less acidic protein. Heterochromatin is genetically less active chromatin and form stop point in transcription. Heterochromatins occur nuclear membrane. Constitutive heterochromatin occurs in all cells in all stages. Example, centromeric regions. Facultative heterochromatin occurs in some cells in some stages. Example, bare bodies. Bare bodies in female cells is a facultative heterochromatin structure. Number of bare bodies in nucleus of an individual in number of X chromosome minus 1. Difference between euchromatin and heterochromatin. Euchromatin consists of a thin, extended light stained part of chromatins. Heterochromatin consists of a thick coiled, condensed part of chromatin and dark stained. Euchromatin genetically more active chromatin. Heterochromatin less active or interchromatins. Euchromatin lie a center of nucleus. Heterochromatin lie near the nuclear membrane. Euchromatin less histone protein, heterochromatin more histone proteins. Euchromatin replicate in early S phase. Heterochromatin replicate in late S phase. DNA to mRNA in euchromatin, DNA to rRNA to tRNA in heterochromatin. Heterochromatin take light stain during the cell division stages, M phase, and take dark stain during the interphase. Chemistry of nucleus. Protein 85%, RNA 10%, DNA 5%. Function of nucleus. Ribosome formations. If the chief role of nucleus, thus it is called as a ribosome factory of cell. The protein of ribosome are synthesized in cytoplasm but is diffused on the nucleus and reach at nucleolus. Here rRNA and ribosomal protein are assembled to form ribosome which move to cytoplasm through nuclear pore. At the some place, heterochromatin form thick dense granules which are known as karyosomes or chromocenter or false nuclei. Function of nucleus. Genetic information. Nucleus contains genetic information in its chromatin. Transmission of genetic information. Nucleus take part in transmission of genetic information from parent to daughter cells or one generation to next. In the cell division, division of nucleus is prerequisite to cell divisions. Control mechanisms. Nucleus control metabolism of cell by sending mRNA in cytosol. Variations. Variation depending due to the changing in the genetical material of nucleus.